So this is, everyone is R, okay? This window over here. And this is the R GUI. And so this is what is known as the R console. So this is where you can make different types of commands or type different kinds of commands. There's also what Dr. Teng was referred to earlier as the R editor. So you can also make, write commands and things in here. And then what the advantage of R editor is that you can save these as scripts. And scripts are, you can just run with a single click. Okay. So so that's the R editor. And then when you start making plots and things like that, then you'll get another window that'll pop up that'll show you graphics. Now, we had mentioned RStudio before, and I'll show you this is what RStudio is. RStudio has the exact same components. So this is the R console. This is where the plots would show up on this tab labeled plots. Can you all see that?
Yes.
Okay. There's also some other helpful tabs like files. So you can change your workspace very easily. So right now I'm in the a directory called workshop. I could easily change my directory to my desktop here, and then I can set that as my working directory if I want to. In R itself, it'll work a little bit differently. The other nice thing about the RStudio setup is that you can see your history really easily. You can also see this in R. There's a function to look at your history. There's also the environment is its own window. So here you would see any variables that you have stored or matrices or what have you. And then up here is an R script window. So you can run scripts from this window here, just as you can in the R editor window here, okay? So we're gonna get started with our handout. So I'm gonna show you some things, and if you have questions, you can ask any questions as I go along, okay? So we'll get started. So the first thing that you'd wanna do, I think there's one thing that needs to be installed for later in this handout. So I'm just going to go ahead and install that now. So I'll show you how to install packages. So you would type install packages, and then you would type QQman. And then a window will pop up basically asking you, to select the crayon mirror. And so it's easy just to select the default. So just do the default, which is 0. And then you'll wait a second. And then as you can see, it's downloaded here. And so all that means is that you just need to copy that path and then go to install packages from local files, copy paste rather, and then it'll take you directly to that window. And then you just select the QQman zip file, and then you select open. And now the QQman package is installed, okay? So I'm gonna go ahead and clear that by typing Control-L. Okay, and so now I have a clean console to get started with. So now I'm gonna show you how to create an object. So we're gonna start by creating object 1. And this is gonna be a list of numbers 2, 4, 7, and 9. And so this arrow, it basically is saying, it's saying that you're going to put this list into an object called object 1, and then you hit enter. Now, if I type obj1, then you see that my list of numbers appears. So don't be concerned with this. This basically just tells you that the first value in this object is 2, 4, 7, and 9, okay? You can, and so I've shown you how to create an object and how to view an object. You can also manipulate too. And so in here, you can see that I've multiplied object 1 by 2. Everyone see that? Then you can also do other things. You can also divide it, subtract, add, all of those things. The next thing that I'm going to show you how to do is how to create a matrix. Okay, so we're going to create a matrix called matrix 1, and we're using this matrix function to tell the R program that we're creating a matrix. And then C again means concatenate. So basically it's just telling it that all of these numbers are to appear together in this matrix. And then we're going to tell what numbers go in the matrix and then what size matrix we're going to use. So the numbers we're using are 1, 2, 3, 4, 3, and 4. And then the size of the matrix is 2 rows by 3 columns. Okay. So when I want to see the matrix, I type M1. And so you can see that we have a matrix that's 2 rows by 3 columns. All right. So then if I just want to view the first row of my matrix, then I just type M1 and then bracket 1, comma, and then close bracket. Okay. If I want to see the second column, then I just type M1, bracket, comma 2, close bracket. Okay. And then if you want to see what's in the cell in the first row in the second column, you easily just write M1 bracket 1, comma 2. And so you can see that number is 3. Okay. There are a number of ready-made data sets in R, and one of those is a data set called trees. So to attach trees, we just type data trees. It's already built into R, so we don't have to go looking for it. And then if we want to see what trees looks like, we just type trees. And so we can see that this is what the trees data set looks like. So it's 31 observations of girth, height, and volume for the trees that they looked at. Okay, so you can also attach trees so that it's just in our environment. And then we can set ourselves up so that we can have multiple graphs in the same window. So what we'll do with that is we'll say par and then mfro. It's basically telling it to make a matrix for our graphics that we're getting ready to make. So par mfro c, we're going to have a 3 by 2 matrix of graphics. Okay. Not sure what that was. All right. Not sure what just happened there. So I'm just going to go ahead and close my R Studio for a moment. And reopen R. Okay. Okay. So again, we're gonna come back to data trees. Actually, we'll just attach trees. And then we'll make our matrix that we're going to use for our graphics. Okay, so it's created this window for graphics. as you can see. And so it's ready for our graphics that we're going to make. So we're going to plot first girth and volume. Okay, so we make a plot, girth, volume, and then we're going to use type B, which stands for both. So, and that means both lines and points. Okay, and I'm going to expand this window. Okay, so as you can see, we've plotted girth by volume. The next thing we're going to look at is plotting, basically plotting meaningful axis labels. So we're going to say plot volume, height, and then xlab. Xlab stands for X label. So that says volume. And the Y label we're going to say is height. And then we're going to give our graph a title, regression scatter plot. And close parentheses. Okay, and so you can see that we've now made a regression scatter plot with meaningful labels. Okay. The next thing we're going to do is we're going to plot a histogram. So in order to do a histogram, you type hist, and then you can type height or girth or whatever. And so you can see now we have a histogram of height. You can also do a histogram of volume. And so you see we have our histogram of volume. We can also do a bar plot. And so we have a bar plot of height for all 31 of our observations. And we can also do a box plot of volume. Okay, so so that is how you make various types of graphics in R. So we can see our window here. Does anyone have any questions before I move on? I think I see someone in the chat. Oh. Okay, no one has, okay, Rashid, sorry.
Yes, so how exactly were you able to make multiple graphs appear in the same window? So when I'm doing it, it only shows one volume, I'm sorry, just
Okay.
one graph in the entire image.
So you see this, you see where I typed par parentheses mfro equals C parentheses 2,3.
Yes.
That is the part of the program to create a graphic matrix.
Oh, okay. Thank you so much.
You're welcome. Anyone else have any questions before I move on? Okay. All right. So I'm going to show you one thing that I think is super useful that I think you'll all want to know how to do. Where is it? So frequently you'll be making graphs in lab, and you'll you'll need to, you'll be making lots of graphs, and you don't want to necessarily have to save all of them individually. So there is a script that I've saved, and I cannot find right now, but, oh, because I'm in the wrong directory, oops. So there's a script that you can use to basically save all your plots. So, if you are, if you've ever made like a whole bunch of plots and you don't have time to save them all individually, you save all of those and it will save them in the directory where you're in your working directory. Okay, and you can come back to them. So that's very useful. Okay, I can share that with you later. In any case, I'm going to go ahead and begin the last part of the handout. So I'm going to clear my window by typing Control L, and we'll start with the GWAS portion. So we've already installed QQman. So now we're going to library our QQman package in. Okay, and now we're going to use the GWAS results. So okay, and then you can head GWAS results. Okay, and so you can see that the GWAS results gives you some SNP information. So these are SNP identities. This is the chromosome. This is the base pair, and this is the p-value. Okay, so you have that information. So the next thing to do is to make a Manhattan plot. So, so you type Manhattan, and then type GWAS results, and then you type CRH. So base pair equals base pair. So this is just telling it where to find this information, where to find the chromosome, where to find the base pair information, where to find the SNP information. And where to find the p-value. Okay, and close parentheses. And so now you've made a Manhattan plot. And so you have two different cutoffs here. So this is like the gene-wide association cutoff here. And this one, I believe you can manipulate, this blue one. Okay. So that's how you make a Manhattan plot. And then you can also subset your information. So so we can look specifically at chromosome 1 if we want. So chromosome 1 subset, GWAS results, comma, CRH equals. Okay. Now we can look specifically at chromosome. All right, so then we can also highlight certain, data. So, so if you wanted to highlight, a particular, SNP or something, you can also do that. Right. Equals chromosome 1. Okay, so, and then main equals Manhattan plot. Those parentheses. Okay, and see, when you make an error, you get unexpected symbol. So there is something I mistyped. Oh, I see where I mistyped. So I forgot to put a quotation mark. And I forgot something else, apparently. Highlight chromosome 1.
It might be the base pair 1.
Oh, yes, I see it now. Thank you. Yes, it is. You are correct. So now you see I've highlighted chromosome 1. Okay. So then the next thing we can do is make a QQ plot. So and then this is based on the p-value. Okay, so okay. Does everyone see what we've done? Does anyone have any questions? Hold on. I don't see any hands raised, so I guess there's no questions. So I guess at this point we can head into breakout rooms. Dr. Teng, would you like to meet those?